Hello, and welcome to today's broadcast, Drug Delivery Nanoparticles Illuminated, the Light Scattering Toolkit Part 2, Online Measurements. This is the second in a two-part webinar series dealing with the application of light scattering to characterizing drug delivery nanoparticles, with a focus on fractionated measurements using SecMiles and FFF-Miles. My name is Lindsay East. I'm the Marketing Manager for Wyatt Technology, and I will host this event. Today's speaker is Dr. Daniel Sohm, Principal Scientist and Director of Marketing for Wyatt Technology. Dr. Sohm has been with Wyatt for over 11 years, first in R&D and then in the marketing department. Prior to joining the scientific instrumentation world, his professional endeavors included the semiconductor and defense industries. Dan completed his undergraduate degree in physics at the Technion Israel Institute of Technology, his doctoral research in the Brown University Physics Department, and postdoctoral research at Los Alamos National Lab and the Weizmann Institute of Science. So without further ado, please allow me to welcome Dr. Daniel Sohm.
Well, thank you very much, Lindsay, for that introduction. Welcome all to the webinar. For those of you who are interested in viewing part one of this webinar series, which dealt with the characterization of drug delivery nanoparticles by unfractionated measurements, it is available on demand at the Wyatt website under Library Webinars. So please feel free to take a look at that. And without further ado, I will proceed to the next part, which is how we can characterize drug delivery nanoparticles with online fractionated measurements. Brief review of part one. Batch, meaning unfractionated measurements, is useful for measuring average values, for instance, average molecular weight, average size, as well as obtaining relatively low resolution size distributions using dynamic light scattering. You can use it to look at temperature and concentration dependence, for instance, determining critical micelle concentration or critical micelle temperature, as well as rapid reaction kinetics, meaning not millisecond reactions, but from seconds up to about an hour or two. The batch techniques include dynamic light scattering, electrophoretic light scattering, and composition gradient light scattering. Composition gradient light scattering is a stop flow technique, so it allows you to make the kinetics measurements as well. Today we'll be discussing the online measurements, or the fractionated measurements, and usually that fractionation takes place by size, by hydrodynamic radius. For instance, GPC, or field flow fractionation based on size. And then we can obtain accurate distributions of size, molecular weight, and other properties utilizing the detectors, the light scattering and other detectors downstream of the separation facility. One of the advantages, in particular for light scattering of online measurements, is that you tend to get cleaner data than in batch, because the fractionation inherently removes dust and large particulates that tend to interfere and generate noise in light scattering data. And if you are looking at slower reaction kinetics, those reactions which are slower than a few hours, it is more common to use the online technique, because you can take you can sample an aliquot every once in a while, run it on the separation. The separation will take typically between 5 minutes and an hour, depending on the particular technique that you use. And you can get high resolution data. And if the reactions are slow, then not much, the sample won't be changing as it goes through fractionation and dilution by very much during that time. The online techniques that are used in the light scattering toolkit include the combination of light scattering detectors with size exclusion chromatography and the combination of light scattering detectors with asymmetric flow field flow fractionation. The reason that we want to apply the light scattering toolkit in research of nanopharmaceuticals is to answer certain analytical questions. For instance, what are the solution properties of the constituent macromolecules, the perhaps polymers of proteins which form the structural aspects of the nanoparticle, the peptides or oligonucleotides, or small molecule drugs which might be the payload or perhaps are immobilized on the surface in order to perform liganded targeting. And the solution properties of those constituents that we'll be measuring include the molecular weight, from which we can, of course, derive a oligomeric state, Also the size in terms of radius, conformation and shape, conjugation state, meaning for complexes which are covalently bound and include, or even non-covalently bound, might include a pegylated protein, a copolymer, or other types of complexes containing two distinct species. What is the molecular weight of each of those species in the complex, and determining the charge of the, or the Z potential of the macromolecules. Techniques which are used to answer these questions include SecMiles, which we'll be discussing today, dynamic light scattering, which were discussed in the previous webinar, and MEPOLs, also discussed in the previous webinar. The next set of questions involve the solution properties of the nanoparticles themselves. So once we've created them and perhaps loaded them up, what are their solution properties? What are the size distributions, aggregation state, their particular conformation or shape? We'll discuss some interesting examples of that. The conjugation state as well, and the Z potential, which essentially measures the charge on the nanoparticle, as important to know in terms of stability. And then finally, once we have a product, we'd like to know if it's well behaved. So does our product aggregate or is it stable? If we are incorporating a drug into a nanoparticle, is it in fact incorporated or is it free in solution? And finally, reaction kinetics, for instance. How does the carrier vehicle degrade when it reaches the physiological environment, or how is it formed? And so light scattering will answer all of these questions for us with a coherent and extensive set of experimental techniques. Brief review of the capabilities of the different flavors of light scattering. So flavor number one is multi-angle static light scattering. And in all the light scattering techniques, we have a solution containing the macromolecules or the particles, which is illuminated with a laser beam. And the light impinges on those particles and is scattered in all directions. In multi-angle static light scattering, we place detectors at several angles relative to the laser beam direction and measure the time average intensity of the scattered light. As we'll see in the next slide, the light intensity actually has rapid fluctuations, but we're going to average over those fluctuations just to measure the average intensity. And thermodynamic theory tells us that the intensity of the scattered light is proportional to the product of the molecular weight of the analyte, its concentration, its weight concentration, and there's also an angular dependence which depends on the size of the particulates. If we're looking at molecules with a radius of less than about 10 or 12 nanometers, there's actually very little angular dependence and we don't actually measure size. But for sizes larger than about 10 nanometers in radius and up to 500 nanometers in radius, multi-angle light scattering can measure size. So from first principles, MALS will measure molecular weight as well as the RMS radius, known as RG or the radius of gyration. That's one way of measuring size. There are other ways of measuring size. MALS can also give us interaction parameters, for instance, the stoichiometry of a complex, the equilibrium dissociation constant, measure of the on and off rates of association-dissociation, as well as measuring non-specific interactions through thermodynamic parameters such as the second virial coefficient, known as A2 or B22. And because it is a first principles analysis of molecular weight and size, it affords in-depth characterization of the solution properties, which is essentially unparalleled. The second flavor of light scattering is dynamic light scattering, also known as quasi-elastic light scattering. And there, instead of measuring the time averaged intensity, we're going to measure the rapid fluctuations in the light scattering signal, which are on timescales of microseconds to milliseconds, depending on the size of the particles. The reason that we see those fluctuations is due to Brownian motion. So the particles will be jittering around in solution due to Brownian motion, and the light wave that impinges on those particles will scatter and reach the detector, and depending on the position, relative position of the particles over time and the distance from the detector and the angle, we might see constructive or destructive interference of those light waves at the detector. And as this changes over the course of their motion, we'll see these associated increases and decreases in the light scattering intensity. By analyzing the rate of fluctuation, we can determine the diffusion coefficient of the particle in solution, dt. And we can use what's known as the Stokes-Einstein equation to convert the diffusion coefficient, the viscosity of the solution, and its temperature to a measure of size known as RH, the hydrodynamic radius. What this gives essentially is the size of a sphere with the same diffusion coefficient as our particles. So from first principles, DLS measures diffusion coefficient and is converted via Stokes-Einstein to the hydrodynamic radius. Hydrodynamic radius is a little different than the RMS radius, and I'll get to the difference between them a little bit later on. Now, one of the nice advantages of dynamic light scattering is that even without fractionation, we can get a rough estimate of size distribution. It's a very low resolution size distribution, but nonetheless, it can be very convenient. If the population is monomodal, meaning clustered around a single size range, then the cumulant analysis is used to give us the average RH and the polydispersity, meaning the width of the size population around that average value. If it's multimodal, meaning that the particles are clustered around a few average sizes, we use the regularization analysis, which gives us rough size distribution from less than a nanometer in radius and all the way up to several microns in radius. And so it's quite extensive in range and quite convenient because it can be done very rapidly. The third flavor of light scattering is electrophoretic light scattering, which is used to measure zeta potential or net charge. In this technique, we will apply an electric field, and a charged particle will drift under that field. We measure the speed of its drift using the Doppler effect. That's where the phase part comes into this analysis, and calculate the electrophoretic mobility by the ratio of the speed of the drift to the applied electric field. That information can be combined with the measured value of RH to calculate the net charge or the zeta potential of the particle. And what Wyatt offers is a more advanced form of phase analysis light scattering called MP-PALS, massively parallel PALS, which offers higher sensitivity, more rapid measurements, reduced applied voltage, and this is particularly important for fragile samples such as biological samples. With additional accessories that Wyatt offers, we can make measurements even of proteins, very small proteins in high salt buffer. We can measure fluorescing nanoparticles and their other unique capabilities that were discussed more extensively in part 1, and we'll see an additional unique capability in this webinar. So in part 1, we discussed 3 different tools. They were DLS, SecMiles, and MP-PALS. Now we'll talk about tool number 4, which is SecMiles. SecMiles takes a typical HPLC system using SEC columns and combines them with the usual downstream UV detector, refractive index detector, which is a universal concentration detector. Even if your sample does not contain chromophores or fluorophores, it can usually be measured using refractive index. And the heart of the system being the MALS detector, which might be a Dawn Helios, which is the higher end multi-angle light scanner detector, or the Mini Dawn Trios, which is the starter level multi-angle light scattering detector. Both of these can incorporate an embedded dynamic light scattering module, which utilizes the same laser and same flow cell as the MALS measurement. And so we'll be measuring molecular weight, size via RMS radius, size via hydrodynamic radius, all in the same volume at the same time, which can make for some very powerful analyses, in particular in terms of conformation and shape. And so this system measures several properties of the system. First of all, absolute distributions of molecular weight and size. Absolute, meaning that these measurements actually do not rely on calibration standards of molecular weight or size. They're measured completely independently as I described in the MALS theory slide. The range of measurement begins at around 200 grams per mole, and depending on which detector you have and your fractionation technique, can extend all the way up to around 1,000 megadaltons, which is quite large, or 10 to the 9th grams per mole. The range of hydrodynamic radii that can be measured using the incorporated DLS module ranges from about half a nanometer. Again, an upper limit which will depend on the specific detector and specific fractionation technique. Using field flow fractionation and the DAWN, you can go up to about 240 nanometers. But of course, SEC doesn't actually separate up to that size range, and so it's limited to about 50 nanometers. RG, again, root mean square radius, beginning with being about 10 nanometers, extending up to 500 nanometers using the DAWN system and FFF. That upper limit would be lower if your separation technique did not extend that high, or if you use the lower end mini DAWN trios. And so this system, this tool, is good for looking at small aggregates, characterizing oligomers, analyzing conjugates, determining conformation from the combination of online dynamic light scattering and multi-angle light scattering. And so why would you want to go to all this trouble of adding on multi-angle and other detectors just to determine the molecular weight when you can do a standard SEC separation or GPC separation, calibrate your column with known standards, and call it a day? Well, let's look at this example. So we're looking at the separation on GPC of two polystyrene standards. So this peak and this peak correspond to known polystyrene standards, and an unknown polymer, which is this blue curve. We have plotted on the x-axis the hydrodynamic radius, which was measured using online DLS. And so what we see is that at any given elution time, the hydrodynamic radius, which is determined by DLS, is essentially identical for the standard and for the unknown polymer. And that makes sense because the separation of GPC or SEC is based on the hydrodynamic volume. And so polymers that don't interact non-ideally with the column and elute at the same hydrodynamic volume should have the same hydrodynamic radius, which is fine. However, if you look at the molecular weight of these standards as determined by light scattering, we see that they are in fact different. And so the molecular weight of the polymer and the polystyrene standard for this one are about the same, but for this one are quite different. And that's because there is not a simple one-to-one relationship between the molecular weight and the size of a given polymer. The elution is based on size, not on molecular weight. And if the polymer differs from the calibration standard, you'll get incorrect measurement in terms of molecular weight. And so it's really important to utilize light scattering if you want to have accurate molecular weights. Accurate molecular weight does matter. You know, you can utilize the wrong tool in your measurement or in your application, and you can see that in this case, it's just not going to do the job. So it's important to make sure that we have the right molecular weight in our polymers. Here's another example of where standard GPC fails. This is a single polymer which was injected with two different injection volumes. So 10 microliters down here, 50 microliters up here. You can see that the peaks of these elute at different times, even though they're exactly the same polymer. And so if you were to use the calibration curve, which is this one over here, you would get different MPs and different MWs for these two different polymers. However, light scattering, measured here for the two, shows us that in fact the molecular weights are essentially identical. So again, here the molecular weight measurement would fail. You'd get up to about 25% discrepancy in molecular weight using column calibration, even though you're injecting the same polymer with just different injection volumes. And light scattering gives you the right answer and tells you that they're essentially the same to within a couple of percent. So why does that happen? That happens because traditional SEC assumes that your calibration reference molecules and your samples are identical and ideal in terms of conformation, density, column interactions, and that your chromatography is reproducible day to day. That is, the elution times don't change. Otherwise, you have to recalibrate and recalibrate and recalibrate. Here's a nice example from the protein world showing where standard chromatography fails. BSA, of course, is one of the typical reference molecules used to calibrate protein columns. And so BSA monomer and BSA dimer elute at different times, and those times, of course, work pretty well in terms of column calibration. The light scattering identifies these two peaks as, in fact, being monomer-dimer through the given molecular weights. Hemoglobin, run on the same column under the same conditions, elutes much later. And so based on standard column calibration, you would expect that the molecular weight of hemoglobin is much lower than that of BSA monomer, but in fact that's not true. Light scattering tells us the correct answer, that it is in fact about 62 kilodaltons. At least the leading edge of the peak is 62 kilodaltons. And the reason that it elutes much later is because it has a different column interaction, even though the size is different. And also, at the same time, there's something else going on with hemoglobin, and that is a rapid equilibrium between the dimer and the tetramer of the hemoglobin. So at 62 kilodalton, we're essentially looking at a tetramer, but due to dissociation during the dilution on the column, there is actually an equilibrium between tetramers and dimers, and we can see that in the molecular weight as measured by light scattering decreasing across the peak, giving us this funny shoulder on the trailing edge. Without light scattering, we would not actually know what's going on in terms of the shoulder. Light scattering tells us the molecular weight decreases, and that's a sure indicator of this equilibrium. Multi-angle light scattering covers a very wide range, as I indicated earlier. And here we're looking at a separation and characterization using moles of both very low saccharides down to a monomer of 214 gram per mole, and up through polysaccharides, which extend all the way up to above 1 million grams per mole. What we see here is injections of different volumes. Now, there's no change, no significant change in elution time, as we saw in the earlier example, and so the molecular weight determined by column calibration would be similar for these three, but light scattering gives us, without need for calibration, such a large range which extends from over 1 million all the way down to 1,000, and then also, if we have enough injected mass, we can actually see the monomer and identify it. So light scattering gives us all masses, large and small, great and small, and is applicable to quite a few different samples, even those which do not have relevant standards on which you can do the calibration. Now I'd like to look at the conjugate analysis for SecMiles, a very important application for light scattering. Examples of conjugates that can be analyzed include pegylated proteins, so a protein pegylated to a carbohydrate, or a protein DNA complex, polymer peptide assembly, or copolymers. The way that we do the analysis is using 3 detectors simultaneously. And the assumption here, it can be done for more complex situations, but I'll describe the analysis for the simplest situation, that is where one of the constituents, say the protein, has a UV response, and the second constituent, say the PEG molecule, does not have a UV response. So the UV detector will give us the concentration of the protein. The T-Rex, which is the refractive index detector, gives us the concentration of both protein and PEG. So between the two of these, we can determine independently the concentration of protein and of the modifier. And finally, by combining the information from light scattering, we can determine the molecular weight of the core protein as well as the molecular weight of the modifier, and get those independently. This is really important for these complexes for which there really are no reference molecules that can be used to calibrate an SEC column. So just here's another way of receiving that. And the results that we obtain are across the peak, the molecular weight of the modifier independently, of the protein independently, and the total mass of the complex. And this, even though this is a simplistic look at how it's done for situations where one constituent does have a UV response and the other doesn't, it can also be carried out for more complex situations where both of them have responses. Besides characterizing just the complex itself, this type of analysis is useful for looking at the results of a reaction where we're looking at a pegylation reaction. And doing the separation, we can identify the unreacted peg, which in this case would have no UV response, the unreacted protein, which in this case would have a UV response, and we would see the molecular weight of the protein, and the pegylated protein. So this is monomeric. Again, we identify the molecular weight of the protein and see that it is monomeric with heterogeneous PEG distribution. And finally, the aggregates, which have multiple proteins as well as many, many PEGs. So to summarize, the tool number 4, which includes SecMiles, you can also add the DLS detector online. I didn't show examples of how that's used, but it can be used to look at conformations. We'll see some example of how that's used with the next tool. We're measuring distributions of molecular weight and size in solution absolutely without reference to reference molecules for calibrating columns. This technique overcomes limitations of standard SEC if you have non-ideal samples or non-ideal conditions, if there are non-ideal interactions between the sample and the column, if there are odd conformations that cause your sample to elute differently than the reference molecules, or if something's going wrong with your chromatography. Can give you positive identification of reacted and unreacted species. And also using the conjugate analysis, we can look at decorated nanoparticles, as well as pegylation, DNA, protein, and other types of copolymers and covalent complexes. If you'd like to look at some additional examples, we have on our website under wyatt.com/bibliography a searchable bibliography of peer-reviewed articles that utilize our instruments to make these measurements. And so feel free to search that and find how your colleagues have been using light scattering detectors to make these measurements. Tool number 5 is FFF-Mols. And so it combines light scattering with field refractionation. Field refractionation is a separation technique that offers certain advantages over size exclusion chromatography, which I'll get to briefly. The Wyatt FFF system, called the Eclipse, integrates with industry standard analytical HPLC systems such as Agilent, Shimadzu, and Thermo Fisher Scientific. We have two modules, and one is called the AF4. The other one is called the Eclipse Dual Tech, which actually allows you automated switching between different modes. So you could use the same HPLC system to run both standard SEC as well as field refractionation, comparing the two techniques, utilizing them in a complementary fashion. The properties which are measured using FFF-Mols are similar to those using SecMiles, molecular weight, size, conjugation, etc., except that they work over a much larger size range because FFF can separate between 1 and 1,000 nanometers. It's very versatile. Briefly, how FFF works: instead of having a packed column, FFF works with an open channel consisting of two parallel plates. The upper plate is just a solid plate. The bottom plate consists of a frit which supports a semi-permeable membrane. And using a syringe pump, using the HPLC pump, we cause the solvent, or the mobile phase, to flow parallel to those two membranes. But we constrict the output, and that forces some of the solvent to flow through the semi-permeable membrane. Now the analyte is too large to go through the membrane, so the cross flow, which goes through the membrane, pushes the analyte against the membrane. There's a counterforce, which is diffusion, which causes the analyte to flow back up towards the center of the channel. The small molecules will diffuse higher. The larger ones will not, and they'll stay closer to the membrane. Now, the channel flow, which is parallel to the membranes, does have a parabolic distribution. So the closer you are to the center of the channel, the faster the flow, and therefore the smaller particles, which diffuse up higher, will come out faster. And so in the case of FFF, the small particles come out first, and the large particles come out later, which is opposite to the way things work with standard SEC. So as I mentioned, FFF is a very versatile technique and can separate sizes from 1 to 1,000 nanometers. It works in an open channel with no stationary phase, and therefore there is essentially no shear, and it is very gentle on the analyte. Because there is so much less surface available for interaction in terms of stationary phase, most of the adverse interactions with surfaces are eliminated. The resolution is as good as or better than standard size exclusion. And one of the really nice things about FFF is that you can optimize the separation for different ranges simply by adjusting flow and geometric parameters. And you can even do this on the fly. And so you can set it up to separate over a very large range, or you can set it up to zoom in and give you high resolution in a particular size range, and then on the fly you can change it to give you high resolution in a different size range. And so it's very versatile in that respect. This is an example of the quality of the separations that you can get with FFF. These are Duke Latex particle standards, and we're using downstream multi-angle light scatter detector to make the measurements of the sizes. And so you can see beautiful separation between particles which differ in size by 30%, 50%, not very much. This is, of course, much, much better than you can get with dynamic light scattering, where we can only if there's no separation, dynamic light scattering will give you populations that differ by a factor of 3 to 5 in size. Here you're getting baseline separation on particles that differ by just 30 to 50%, and we'll actually see an example later on of even better separation. Another thing to note in this fractogram is that the light scattering gives you the polydispersity or heterogeneity of those Duke Latex particle standards, which you would otherwise be hard-pressed to know about. And you can see that the heterogeneity is actually pretty large for these 25 nanometer particles, and it's very good for the 150 nanometer particles. So if you need to characterize your reference standards, this is a great way to do it. It gives you excellent accuracy and precision in the analysis of these particles. And so, as I mentioned earlier, we can look at even better resolution. And this is an example of separating virus-like particles. And there are two different particles that correspond to 3.4 megadalton and 4.7 megadalton as measured by downstream light scattering. And the particle size are 16 nanometer and 18.5 nanometer, and there's nearly baseline separation. There's very good separation between them, even though they differ in size by about 10%, a little more than 10%, 15%. This type of resolution would not be feasible without size-based separation. If you used a batch DLS or nanoparticle tracking analysis, it would be impossible to get this degree of resolution, not to mention the fact that you can get the molecular weights very accurately using downstream MALS detectors. And if you have fluorophores, you can put on fluorescence detectors downstream, UV detectors, and anything else you need in order to characterize the different particles. One of the ways that FFF is used in drug delivery nanoparticles is characterizing the formation of virus-like particles. And in this example, we show separation between a well-formed virus-like particle with a radius of 20 nanometer and a molecular weight of 17 megadaltons from some incomplete fragments. So the first peak here with molecular weight of 2.5 megadalton was incompletely formed. and the process was not complete. This was borne out very nicely through the separation. The same type of conjugate analysis using 3 detectors that I described in the context of SecMiles can also be used with FFF-Miles. And this is an example of a virus-like particle with a payload of DNA. Again, using the 3 detector technique, we can determine independently the molecular weight of the payload, which was about 1 megadalton, the protein, which was 14.5 megadalton, and of course the total molecular weight being the sum of those, 15.5 megadaltons. So this is a really good way to verify that your payload is in fact encapsulated in the delivery vehicle. This is a different type of, very different type of drug delivery vehicle, a nanolipid complex. And in this instance, the researchers wish to study the binding of microsilver particulates to the nanolipid complex. And they looked at different conditions that led to different degrees of binding of the microsilver. So different concentrations of the nanolipid and different concentrations of microsilver. And these were the 4 different conditions. Okay. And finally, took those samples after they'd incubated and ran separations on field flow fractionation with multi-angle light scattering downstream. Now, I'd like to note that using batch dynamic light scattering and NTA, they weren't able to determine any difference between these processing conditions, but with the exquisite separation capabilities of FFF plus downstream light scattering, it is possible to discriminate very nicely and very repeatably between these different conditions. And we can see here the different sizes of the molecules. So this is radius using multi-angle light scattering. There's, of course, a very large distribution, a large heterogeneity within any given processing condition, but between the 2 conditions, there's a significant difference. The precision repeatability of these measurements are just a couple % in size. And so the differences that we see here, which are on the order of 10% or more, are quite noticeable. If we combine online dynamic light scattering with multi-angle light scattering, it's possible to determine what's known as ρ, the shape factor, which is the ratio of the root mean radius determined by multi-angle light scattering to the hydrodynamic radius determined from dynamic light scattering. And what we plotted here is a theoretical plot corresponding to the expected value of ρ, which varies from 0.8 to 2.4 in this plot, for ellipsoids, which have a minor axis of 1.5 nanometers and major axis ranging from 2 to 100 nanometers. And so those different aspect ratios will give us different values of ρ. And here are 2 different examples of separations of nanoparticles. They elute at different times, but that's not enough to really tell you anything about their shape. But by measuring simultaneously the moles and DLS, we can calculate ρ and find that the first nanoparticle corresponds theoretically to a solid sphere, and the second one is rod-like with a bit of a distribution in terms of aspect ratio across this peak. So it's a good way for determining size. Liposomes are, of course, one of the most popular vehicles for delivering drugs in a nanoparticle system. And it's really important to be able to know if the liposome is encapsulating, in fact, the active ingredient, the drug. Now, if you measure empty or filled liposomes using dynamic light scattering, if those liposomes were prepared in the same way, they will have the same size. The reason is the dynamic light scattering hydrodynamic radius essentially measures the envelope of the particle. So the envelope would be the size of the external surface, and for these 2 liposomes, the envelope is the same, regardless of whether they're filled or not. Dynamic light scattering gives essentially the same value. By that, and here this means batch dynamic light scattering, so there's no separation going on. You don't see any difference between them. And so what we like to do is find a way to discriminate between those liposomes which do or do not carry a payload using combination of the online separation with field fractionation, combining dynamic light scattering with multi-angle light scattering. Again, if we do the separation of these liposomes, the empty liposome being the red and the filled liposome being green, and measure the hydrodynamic radii, they're identical. And that's because the external or the envelope of the liposome populations are the same. If we measure them with multi-angle light scattering, the size, the RG, the root mean square radius, is different between these two. And the reason for that is shown in this slide here. So how do we interpret these radii? RH, as I mentioned, is the envelope. RG, the root mean square radius, is actually weighted by the mass of the particle. And so if we have a uniform sphere, theoretically, the RG would be square root of 3/5. Sorry, that should not be under the square root. It should be a square root of 3/5 of the radius of the envelope of the sphere. And so the ratio of RG to RH, or rho, would be 0.77. On the other hand, if we have a hollow sphere where all of the mass is located in the envelope, then RH and RG will be identical, and the ratio will be 1. Okay, now notice that we got same RH for these two liposomes, filled and empty, but different RGs. And if we plot those in terms of RMS radius versus hydrodynamic radius for the two populations, we can see that the red population, which is the empty liposomes, fall in a line with a slope of exactly 1, meaning that and this tells us that they are in fact empty, whereas those that are filled fall in a line with a slope of 0.77, which matches the theoretical value for a filled sphere. So we have nice discrimination between them. Now, one of the questions that often comes up in the context of this example is, how do we, if we have a mixed population of filled and empty liposomes, can we characterize them? And the answer is, because the separation is based on RH, the hydrodynamic radius, those two populations will not separate. In other words, you'll not have the filled populations occurring in a different elution volume than the filled and empty will not occur in different elution volumes, because in terms of their hydrodynamic volume, they're identical. What you will see is an intermediate value of rho between 0.78 and 1. And so that will give you an indication of how many are filled and how many are empty. Okay. So in this case, by combining mols and DLS downstream of FFF separation, we can learn structural information on our nanoparticles. And that structural information is critical. Again, in this case, you wouldn't want to be employing a particle which is filled and has much larger molar mass than what you expect. Another application of FFF mols is counting the number density of nanoparticles. And so what we're looking at here is adenoviruses. This peak corresponds to the monomer, and then there's a little shoulder over here which corresponds to dimers. And so we can determine, of course, their radius. But if we know their radius and we know the refractive index of the nanoparticle, it's possible to calculate how much light scattering intensity each particle will generate. And so by dividing the total scattered intensity by the individual intensity from each particle, we can determine how many particles there actually are in the scattering volume. And we've done that for, this publication did that for these adenoviruses. They also counted them using an imaging technique, electron microscopy or force microscopy. and found essentially the same values in terms of the average radius of the particles and the number density of the particles, difference of just a few percent. And one of the advantages, of course, of FFF, besides being a lot faster, is that you're sampling a much larger number of particles than you typically do using an imaging technique. So if you're using electron microscopy or other imaging techniques to characterize these nanoparticles in terms of size and size distributions, you're missing many particles. Whereas the FFF has a relatively high sampling efficiency, and your ensemble is much larger for more accurate averaging. So I'd like to look at another application of this same number density characterization. And this was the characterization of microgels, which are coarse shell microgels, and involve some degradation by breaking links on the shell. Okay. So we had a core with a different type of shell, and the reaction caused the degradation of the shell, which caused change in particle size. And what we did was, or what the group who did this study did, was react their particles with the scission chemistry, run separations on FFF, and look at changes in size distributions. And you can see that we have an exquisite separation here between 52 to 53 nanometers, 54 to 56 nanometers, and 57 to 58 nanometers of the core particles, the non-degraded core shell molecule or particles, and the degraded core shell molecules, which differ depending on the particular chemistry that was used and the types of shells that were implemented. They're just very small differences in size, but they're clearly established using this technique, and the distributions are very accurate. So you get excellent accuracy and precision in terms of looking at the very small changes occurring in these chemistries. Finally, we can take FFF, and instead of using multi-angle light scattering or dynamic light scattering downstream, we can incorporate the Pals detector. So the Mobius, which is Wyatt's MP Pals detector, measures electrophoretic mobility, utilizes a flow cell and can be run in flow mode. This is an example of looking at separation and measurement of the electrophoretic mobility of a mixed nanoparticle species with 50, 100, and 200 nanometers. These are latex spheres. And so we're able to measure the size because the Mobius incorporates a dynamic light scattering detector, which will give us the hydrodynamic radius. I'm sorry, that's the red, RH, as well as the electrophoretic mobility measured here in blue. And so if you would like a better characterization than what you get in batch in terms of electrophoretic mobility of a mixed population, you can do this online separation with immediate downstream measurement of the electrophoretic mobility and size using the Mobius. So to summarize, FFF-MALS, tool number 5, is the primary tool for fully characterizing the size and conformation of nanoparticles and submicron aggregates. It's the primary tool for characterizing complex fluids, which might consist of macromolecules and submicron particles, free and bound or encapsulated drug. It's the primary tool for molar mass inside of proteins and other macromolecules that do not elute well on SEC. So if you have adverse column interactions, the macromolecules are sticking to the matrix in ways that prevent good characterization. Often FFF is a better way to do the separation and characterization. It's utilized for vesicle validation analysis, and you can even incorporate a downstream online zeta potential measurement using the Mobius. We have coming in the future an additional separation which incorporates electrical separation in addition to asymmetric flow separation. So you'll have separation which separates by size and charge. Downstream of that, you can incorporate the detectors which will give you the very accurate measurements of the size and charge. So we have even more coming in the future in terms of capabilities for this tool. So to recap, the questions that we asked at the beginning of our particles and constituent molecules: what are their solution properties, and how do they behave, how do they react? We can see that we can get very nice characterization of many properties in solution using light scattering techniques simply by combining the right type of sample prep and delivery with the right type of dynamic light scattering or multi-angle light scattering or phase analysis light scattering detector. Often we can do those in combination and get even more information, for instance, conformation, by combining multi-angle and dynamic light scattering. If you're interested in exploring some of the ways that our light scattering tools have been utilized in the literature, you're welcome to do a search in our bibliography, which has over 11,000 peer-reviewed publications citing Wyatt instruments in the research, including many from Nature, Science, PNAS, Cell, JACS, etc. And if you have any questions about the information contained in this webinar, I welcome you to email me, dsum@wyatt.com, as well as to browse our website. We have sections dealing with solutions for different types of applications. We have an extensive library of theory, application notes, including the on-demand webinars, which cover a lot of this material in different ways. There are webinars which delve more deeply into the theory of dynamic light scattering, multi-angle light scattering, different ways of doing characterizations. And so I invite you to browse those as well if you'd like more information. And with that, I've concluded this webinar, and we'll turn it over to Lindsay.
Great. Thanks so much, Dan. This now will conclude the webinar. So if you have any questions regarding this webinar, please email Dan at dsome@wyatt.com. We'll also be posting a running Q&A summary on the webinar's webpage with the questions and answers. Wyatt's on-demand webinar library is a great resource where you can find recordings covering many additional aspects of light scattering technology and applications. Please look for the first webinar in this series, Drug Delivery Nanoparticles Illuminated, the Light Scattering Toolkit Part 1, Batch Measurements, which focuses on unfractionated measurements using DLS, MP-PALS, and SecMiles. Thanks again for joining us, and this now concludes our webinar for today.